Welcome everyone to This Week in Science Education, episode number 88. Our topic for today is sexual selection. I'm Brent Peltola. I'm sitting in today for our regular host, Kevin Kugler. I'm joined today by our co-hosts, Thomas Merritt from Laurentian University and Colin Jago from Kawartha Pine Ridge District School Board. We're pleased to be joined today by our guest, Lindsay Derraugh from Carleton University, who's doing work in the evolution of reproductive strategies. Lindsay, I wonder if we can ask you to start by telling us something about your research.
Sure, thanks for asking. So I am a current PhD student at Carleton University. I am an evolutionary theorist in the strictest sense in that I develop hypotheses rather than a traditional scientist or most types of scientists that test hypotheses or develop methods to test hypotheses. I'm the first stage in that process where I actually develop the hypotheses. So a couple main questions that I ask that maybe we should talk about today, or that is related to the subject of sexual selection. One question that I ask is, are there fundamental differences between males and females? Not just on the diploid level, which means individuals with two sets of chromosomes, which translates roughly to adults or babies or children, not just those subset of individuals, but I specifically deal with eggs and sperm, so single-celled individuals. And I do call eggs and sperm individuals just for the purpose of my research. A second question that I ask is, can sexual selection occur within an egg cell? Which means after egg and sperm have come together, is the female or the male still able to make a choice whether or not they want to mate with the other individual? That's a brief overview of my research, and maybe we can start with that.
So I have a question just at the conceptual level. So you're generating hypotheses. I went in and looked a little bit at your research, and I looked at your advisor's webpage. And you're very explicitly separating testing empirically the hypotheses from generating the hypotheses. So how do you generate a hypothesis without an intent to test it? I'm missing a step there, and this is something I think that it would be interesting for the students to get an idea conceptually how your research differs from somebody who's more of an empiricist like myself.
Yeah, that's a great question, and I get that question all the time from everybody. So what I do, that's different. So I basically see it this way. I can break science down into three parts. You have the hypothesis building, which is what I do, which theorists do. Then you have the method, which is doing things like building mathematical models or running simulations, stuff like that, building the tools to test it. Then we have the vast majority of scientists that actually test hypotheses. And my goal is to have my hypotheses testable within the next 10 years. One of the reasons that I develop hypotheses and not actually test them is because some of the hypotheses that I develop are not testable at this point in time. But I always strive to be realistic in my research and to have something be testable sometime within the near future.
Sorry, let me just follow up on that. I'm sort of taking devil's advocate here, mostly because I'm an empiricist, and so I do the other end of the spectrum.
Right.
How does what you do differ from just making things up? So and I mean this in a very constructive way, and as I'm putting this together in my head, but it sounds like you're making stuff up, and somebody else is going to test it, and you're obviously not. So how is this different from just making stuff up?
Well, in a way, I'm just making stuff up, really. What I do is I tend to read a lot. I try to do as much as possible. I try to synthesize ideas as much as possible, and I do a lot of thinking. But really, I am just making things up. I'm making educated guesses of what I think is going on in certain situations which have not been explained in the past.
I think that the thing that for students to take away from this is that you're not just making it up. You're not pulling this out of air. It's based on the work that's out there. So maybe give us an idea how you do that. How do you go out and see what has been done and synthesize that into hypotheses? And then I'll shut up and let Colin ask a question.
So, like I was saying before, I read a lot. I tend to keep up with new journal articles that are posted all the time. I'm constantly reading. I'm making spreadsheets trying to carefully catalog information at all times. I do a bit, not a lot of modeling, but I will plot ideas on trees of life, which are called phylogenies. And I bounce ideas off other individuals. I go to a lot of talks. I talk to a lot of people. I look at the oddballs. So I look for things that go against the trend, and I ask the question, well, why is this one an outlier? What's going on there? Rather than look at the trend.
I read a lot about science and stuff as well, but a theoretical physicist I can wrap my brain around, right? But theoretical biology is something that I've not really ever considered, so this is really cool. So in terms of developing hypotheses, I think about something in physics like the Large Hadron Collider. They came up with a hypothesis. They said, we don't have the technology yet. Let's work on the technology. What would be the kind of hypothesis in biology at this point in time that we think we want to test, but we don't know how to yet, or we don't have the technology yet, and how do we get to that point where we can test some of these things?
So an example would be one of the hypotheses I've proposed, and that is the hypothesis of sexual selection within an egg cell. So I've proposed that this phenomenon can occur. So once egg and sperm come together, there can still be some selection that goes on within the egg cell. So the female is not always necessarily going to mate with the male just because the male sperm has entered in the egg. She can still reject that. And this is not, so let me step back a bit. So I have two requirements that must be met in order for sexual selection within an egg to be possible. One is that the sperm have to be able to act independently within an egg cell. So humans are odd in that they only have one sperm that fertilizes the egg. But a whole suite of different species have multiple sperm that fertilize the egg. Sharks, for example, can have hundreds of sperm fertilize their egg. So how does the female decide which sperm to fuse with in that situation? So I'm proposing these are the situations that sexual selection can occur, as long as two requirements are met. One is that the sperm need to be able to behave independently. So they have to be able to not be controlled by the egg cytoplasmic factors or controlled by the egg. They have to be capable of independent action. Okay. And they also have to have some sort of different defining features. They have to be different from each other. That's an example of something that we're not able to test at this moment. We don't know. We have some ideas of how these sperm may differ. We're not exactly sure what they are, and we're not able to test it at this time. We can make observations, but we just don't have the methods to test those kinds of questions. So that's an example.
Okay, that's a good example. In your work, like I'm thinking now of the kinds of things that scientists write and publish. Would you, as you propose a hypothesis, propose maybe a possible, I know you said you're not into developing methodology and technology, but would you propose a possible way of testing that in order to drive maybe those who would go down that road or in the future? Like, is that part of your thinking as you write and publish these things?
If I know a way that it can be tested, for sure, I will put that right out there. and I might even find a way or see if I could find a student to help me test it. But really developing ways to test things are the methods portion of science, which I don't really do as much.
So let me jump in just for a second. I have a specific question about exactly what, about your research and from the sort of odd biological cases perspective. But Colin, as a quick example of how other people do this, it was either Watson or Crick. We'll say Watson. You can check me and find that I'm wrong. Proposed that to get from the DNA to amino acids, there needed to be an intermediate molecule. And when he made that proposal, we didn't have a way to test that, right? But decades later, we did, and we came up with tRNAs. And I've read some bits and pieces where it was either Watson or Crick said the most intelligent thing they did was propose that bit that we couldn't test at the time, right? And that was sort of pushing the envelope of what we could do because we didn't have the tools, but intellectually we could put that together. So I think this basic idea is really neat, and what you're getting at is asking questions that push us to find ways to answer them, and that's awesome. When you're talking about the fusion of the sperm and egg and the independent roles between those, one of the things that came to my mind was parthenogenesis. And there are multiple species of lizards that are essentially unisexual and have females who give birth to young. In at least one group that I can think of, they actually mate, and the function of the sperm is simply to trigger the egg to go through the rest of development. And the sperm itself is not incorporated into the egg. So is this the kind of odd case that you're talking about when you look at the oddballs to try to inform the questions that you're asking?
Yeah, those are the exact type of things that I look at. Parthenogenesis, especially in lizards, is a really interesting case. I also look at two other phenomena called gynogenesis and androgenesis, and I can explain them. So what gynogenesis is, and it occurs in 7 different phyla, I believe. Occurs in fish, possibly some birds. Those are about the only vertebrates, but a whole suite of other species. And how that works is an egg will be fertilized by a sperm, but instead of the egg and sperm DNA coming together afterwards, the egg will eject the male DNA and then reproduce parthenogenically, like Thomas mentioned, like the lizard. Androgenesis is the exact opposite. It's fairly rare, it's extremely rare actually. It occurs in about 3, genera. Bacillus insects, and it occurs in mollusks, and it occurs in a species of citrus tree. And what occurs there is a sperm or multiple sperm will fertilize an egg. And in this case, the egg DNA is ejected, and it's either 2 sperms come together to form a new individual, or 1 sperm will duplicate its chromosomes to form a new individual. So these are the types of things that I look at. I look at these weird forms of reproduction or these weird oddball cases and try to figure out, well, how come these are like this?
That's awesome. I mean, one of the other weird examples that I know of in fusion of gametes is in mollusks, all mollusks, some mollusks. The normal pattern where the only mitochondria that make it to the next generation, or in the female, is not followed. And in different tissues within a bivalve, it's the male mitochondria or the female mitochondria, and I may be getting some of the details wrong. But with the work that you're looking at, do you go beyond the nuclear genome and into things like the organelles?
Yes, we do actually. Yeah. So you're right. Mollusks are very interesting. Mollusks have what they call double uniparental inheritance.
Yeah, that's awesome.
Big slew of words, but it means that they actually inherit their mitochondria from both parents at the same time. A lot of plants actually do not follow maternal inheritance of mitochondria as most people expect they would. They take them from one or the other. So I do look at things like that. I also look at centriole inheritance, which generally comes from the male. It's paternally inherited. But in species such as carcinogens or in gynogens, where they don't have the input of those male organelles, I look at, well, where are these organelles coming from? Why is this different? Can we apply this to anything else?
That's super. Colin, how does this possibly tie into high school classes, other than we're talking about sex, so the kids are going to be interested?
Well, that's every high school class right there. You've got them hooked once you start talking, right? I'm actually sitting here thinking about the evolutionary connections, and we're talking about all these oddball sort of examples, and you're generating hypotheses. Does this then help inform, say, hypothesis in the past as well about the evolution of sex in general? Because that's totally something that connects with high school curriculum. Is that the kind of thing that, and we're talking about hypotheses going forward, but does it also then help to generate hypotheses about what has happened that people are maybe exploring and testing?
So for my research defining the differences between males and females, most individuals would say, well, we can say that eggs are large and non-motile, sperm are small and motile. Well, I use oddball examples to throw that back in the face of everyone and be like, hey, no, look, we see very large sperm in Drosophila flies, for example, which are fruit flies. Or we see non-motile sperm in this weird example of elephants. So I try to deconstruct other people's hypotheses and definitions as well as a part of my research, which probably isn't appreciated all the time, but I think it's got to be done sometime.
One of the things in the examples, like the example you just mentioned of some of the Drosophila with the enormous sperm, those are usually seen as derived cases and not ancestral. So we tend to downplay the importance of those because it seems to be something that's evolved along an unusual trajectory. It's not something that we see as a common ancestor. Does that play into the way that you're looking at the things that you use to generate hypotheses?
In the case of the Drosophila, I'm not really sure. It's quite possible that it is derived. I don't have a clear answer on that one. But often, individuals just assume that the oddballs are derived, which they might very well be, but sometimes they're not. And even if they are derived, it still begs the question of, well, why? And does the exception prove the rule or not?
And that was sort of getting to the point I was going to make, was that all of these exceptions to the rule at some point need to come under some sort of model that helps explain some of these exceptions, and that's exactly what you were getting at there. So yeah, and again, that's a process of science understanding, right? And that's something that we really try and help high school students understand. Very often we talk about oddballs to help explain, general patterns, but then, these are outside. I mean, it all fits together. So yeah, that's cool.
That's great. Man, I think we're running out of time. I can't actually see my clock from here, but can I ask one last question? Of all you're talking about all these oddballs, what's the craziest example out there that you've run into? I mean, you've got to have had this question before. So what's the most wacky one out there that helped you redefine the way you were asking questions?
Well, what started me on this whole thing several years ago now was gynogenesis, was the example of when the sperm enters the egg and then the egg ejects all the DNA. So that's pretty crazy. Another crazy example which started me on a different tangent was from a species of comb jellyfish, where they're polyspermic, which is, like I was talking with the sharks, so multiple sperm fertilize an egg. And there was one obscure study done back in the early 80s that they actually took a video camera and videotaped the behavior of the female nucleus, the brain, as maybe we can call it for this case, of the cell. And what it was doing is it was migrating to different male nuclei within the cell before fusing with just one. And they had a videotape of this, and they traced the trajectories, and that was just really crazy too. So that's maybe my second most crazy thing.
That's awesome. Thank you.
Well, Lindsay, I think we are getting close to the end of our time. Really want to thank you for an incredibly interesting topic today. Fabulous discussion. So thank you for your time. Thank you, Thomas and Colin. And have a good day and end of episode 88. Thanks a lot, guys.